Good morning, biology students. So we are moving into hopefully the last section that we'll have to do this way. Hopefully when we get back from spring break, I will be there to be teaching you guys in person. It might be 4 days a week, 3 days a week. We'll see, but I should be back. So I hope it's the last time I actually have to record this. Cellular respiration. So this is the process that goes hand in hand with photosynthesis. So you have photosynthesis, which is what makes glucose through from using the sunlight, carbon dioxide, and water. And then cellular respiration is what actually takes the products of photosynthesis and uses them to give us energy to use for all the processes that our bodies go through. And the amazing thing of cellular respiration is that the very reactants that it needs are the very products of photosynthesis. And the products of cellular respiration are the reactants plus light for for photosynthesis. So it's this beautiful, beautiful cycle. So we're going to do something like we did with photosynthesis. I'm taking, this only is 2 sections, and we're going to put together, and then we're going to touch base on what's called fermentation after this real quick. I don't even know if I'll fully test on it, but fermentation, you just need to know, is the process of making energy without oxygen, pretty much. Okay, so we'll talk briefly about that because you will need to know that on the end of course exam possibly. So we're going to talk about that just briefly after cellular respiration. So we're going to just do these 2 sections and then have an assessment. So key questions: where do organisms get energy? What is cellular respiration? What is the relationship between photosynthesis and cellular respiration? Just kind of gave you a quick synopsis of that. And then how much ATP does cellular respiration generate? So you saw that ATP was used in photosynthesis, but it just happened to kind of be there at the disposal of, the cell. But where does it actually come from? This is the process that it comes from. Okay, so when you get hungry, how do you feel? Kind of think about that. Like most of us, you probably feel a little sluggish. You might get a little dizzy and you feel weak. Like if you haven't eaten, like if you ever watched any of those like survivor shows or where they're like put in the wild with nothing, sometimes literally nothing. What happens to them? They get tired, they get dizzy, they get weak, and so on. And that is very, very normal. Okay. You feel weak because you are hungry, because food gives you this source of energy that your body needs. This is your body's way of telling you that your energy supplies are low. That's why God created these hunger signals that say, hey, I need something, I'm missing something. And then it's our responsibility to then, put energy into our bodies. But how does the food that we put into our bodies get converted into energy that we can actually use? Okay. So our bodies burn energy just like a car burns the gasoline we put into it. So food provides living things with these chemical building blocks that we need to grow, we need to reproduce. And the food molecules contain chemical energy that is then released when the chemical bonds are broken. So organisms get the energy they need from food. So organisms that are heterotrophs have to actually consume food. Now, obviously, a plant, there are some carnivorous plants And there are some organisms that are more, that are kind of like stuck in place versus us who can go to our refrigerator. So how we get our food and where we get our food may vary from organism to organism, but we still need to get that food. But how much energy is actually present in our food? Well, there's actually a lot of it, but it just kind of varies with the type of food. Energy that is stored in food is expressed what we call units of calories. A calorie is the amount of energy that's needed to raise the temperature of 1 gram of water 1 degree Celsius. So that's all a calorie is the amount of energy that is needed to raise the temperature of 1 gram of water. That's not a lot of water. That's a very tiny bit of water. 1 degree Celsius. Not 1 degree Fahrenheit, 1 degree Celsius. And the calories on food labels are actually kilocalories, which means they are 1,000 calories. So if you are looking at a food label and it says it has 25 calories, it actually is 25 times 1,000. Okay. So it's actually 25,000 calories. But all calories are, it's just, like I said, it's just a unit. A lot of times we use them. We've talked about this a lot in science class, where like one term that we use in science is different than what we use in like normal everyday life. So the calories that we're referring to in science, it's just simply energy, the amount of energy needed to raise 1 gram of water 1 degree Celsius. Okay, so that's it. And different amounts, different biomolecules, the energies in different biomolecules are going to vary. So a gram of glucose, so glucose is the food source, the carbohydrate that is made in plants through the process of photosynthesis, releases 3,811 calories of heat. Remember, heat is when the molecules touch. It's that. But a gram of beef fat, so there's the fat, releases 8,893 calories of heat. So you can see that various foods have different amounts of calories. Okay. In general, carbohydrates and proteins pretty much have about 4,000 calories or 4 calories of energy per gram. Okay, so remember 4,000 calories, but if it were to be on a food label, it would actually say 4 calories. Whereas fat is going to be closer to 9,000 calories or 9 calories per gram. Okay. So, but of course, cells don't simply burn food and release energy as heat. Instead, they break down the food molecules just really gradually. They capture little bits of chemical energy every step along the way, all these processes. They don't do it all at once. As a gradual thing. And so this really enables cells to use the energy that is stored in different foods, like glucose, which is really used to synthesize ATP. So glucose is so important because it is what is actually allowing the cells to produce ATP. Okay. So here's a quick overview of cellular respiration. This equation probably looks really familiar to you because if you just flip-flop it, you have photosynthesis. So cellular respiration is the process of energy conversion that releases energy from food in the presence of oxygen. This is key, oxygen. Okay. If we don't have oxygen, then it's fermentation. Okay. So the equation for cellular respiration is 6O2 plus C6H12O6 equals 6 carbon dioxide plus 6 water molecules plus energy. So the difference is we need, so energy is always going to be with the carbon dioxide and water. In the photosynthesis part, it is light energy that's used to make this. And in this case, these two are used to actually make ATP energy. So the words are glucose plus oxygen plus glucose equals carbon dioxide plus water plus energy. So that's what happens. This cell has to release the explosive chemical energy and food molecules a little bit at a time. And it's trapping that energy in the form of ATP. Remember, ATP is an adenine triphosphate. Okay? Adenine triphosphate. Tri meaning 3 phosphates. ADP is adenine diphosphate that only has 2 phosphates. So as you can see, cellular respiration requires oxygen and a food molecule such as glucose. Now, can it use other molecules? Yes, it can use other molecules, but glucose is the most efficient and helps create the most energy. But yes, you are still going to get energy from other food biomolecules, but glucose is the one that's the most efficient. That's just the process that God had made. And then it gives off carbon dioxide, water, and energy. Because what do we breathe out? We breathe out carbon dioxide. There also happens to be water in that when we breathe out. And we also happen to go to the bathroom, which is also water that comes out of our bodies. Okay? So, but do not mishear me here. This looks really simple. Okay, but it does not happen in just one step. So like photosynthesis happened in 2 steps. There are multiple steps to cellular respiration as well. Multiple steps, okay? So there are stages. There happens to be 3 stages of cellular respiration, not just 2. Photosynthesis takes place in the chloroplast. Remember, the light-dependent part of photosynthesis takes place in the thylakoids. And then the light-independent portion takes place in the stroma, which is all a part of the chloroplast. But in this case, we have 3 processes: glycolysis, Krebs cycle, and electron transport. Look, electron transport. We did see electron transport chain as part of photosynthesis inside the thylakoid. Okay. Where do you think this happens? What organelle, so stop and think about it. What organelle do you think cellular respiration takes place in? I'll give you a second. See if you know it. Mitochondria. Because what do we call mitochondria? The powerhouse of the cell. That's because ATP, the energy the cell needs, is made in the mitochondria. Okay. So, cellular respiration captures the energy from food in 3 main stages: these glycolysis, Krebs cycle, and the electron transport. So, although cells can use just about any food molecule for energy, so they can use just about, we are going to concentrate on just the one example of glucose because it is the simple sugar and it's the one, again, that is found in all plants. Glucose is found in plants, and it is what the chloroplasts make. So it's really simple. But yes, you can use other forms of a food source there. But the equation is going to look a lot different. Okay. So you can't, it's not the photosynthesis cellular respiration equations that so beautifully balance each other out. Your cellular respiration would just have a different molecule going in, and so on. That's kind of the beautiful thing, is that there are these macromolecules, right? Like our carbohydrates, our proteins, our fatty acids, and our nucleic acids. We don't really digest nucleic acids a whole lot as like an energy source, but carbohydrates are made out of the same things. Carbohydrate, remember, carbons, hydrogens, and oxygens. So it just means that they have a different amount of carbons, hydrogens, and oxygens than glucose, which means that yes, it can use it. It just isn't quite as efficient, and so on. Okay. So glucose enters our chemical pathway. Our first step is known as glycolysis. And only about 10% of its energy is captured here in this stage. About 90% of that chemical energy that was available in glucose is still unused at this point. Okay, so I want you guys, if you don't have your textbook open already, I really want you to open it up to page 312. Okay, page 312. There is an image up there, figure 10.1, that is showing you the three parts. Glycolysis, Krebs cycle, and electron transport. Okay, so you guys can kind of see that. So glucose first enters into glycolysis, okay? And you can see that usually the black is a carbon. The little black circles are going to be carbon. And you know that glucose has 6 carbons in it, right? 6. So this is actually broken down into 2 sets of 3. Okay. Then we have something called pyruvic acid that enters the Krebs cycle. So you're like, where did this come from? Well, it's the 90% that's still unused. That's referred to as the pyruvic acid. So glucose enters glycolysis. Only 10% of its energy is captured to produce ATP during this stage, and the rest of it is referred to as pyruvic acid. So, and there's a little bit more energy that's captured here too. Okay, but most of the energy that comes from cellular respiration is going to happen in our electron transport system. Okay, the electron transport uses oxygen. Oxygen is the world's most powerful electron acceptor, the most powerful electron acceptor. Okay. You're like, why is that important? I'll tell you in a second. So, anytime a cell needs energy, its use of oxygen increases. Okay. Oxygen is required at the very end of the electron transport chain. Anytime a cell's demand for energy increases, its use of oxygen increases too. Think about that for a second. Just stop and think about that logically and apply it to your life. Some of you are athletes. Some of you play instruments. What happens when you start running more or playing more? What do? You start breathing heavier. You are taking bigger breaths. You are breathing faster because your body is using more energy, which means you need to bring it into your body a little bit more. Okay. If you play like a wind instrument, before you go blow on that wind instrument, you got to take a really big breath because you are going to blow a lot of out. And as you play, again, your heart rate increases as you're playing faster. And so your body is requiring more oxygen. That just makes sense. That is just, I said, just thinking about it. If you go hiking or you're a swimmer, anytime you put your body and you start using it at a higher level than baseline survival, you are going to require more oxygen. Okay. So most of these energy releasing pathways within cells are going to require oxygen. And that is the reason we need to breathe. We have to breathe so that we can give oxygen to our body so that it can power our body, so that it can breathe again. Okay? So, pathways that require oxygen, such as the Krebs cycle and electron transport, are referred to as aerobic. Yes, it sounds like aerobics because when you do aerobics, you need to do what? Breathe. Okay, you need oxygen into your body. There's a reason certain words are used for certain things, guys. Okay, so aerobics probably got its name from something like this, aerobic, which requires oxygen. Okay, so, but hear me out. The Krebs cycle, though, doesn't require oxygen directly, but it's classified still as an aerobic process because it cannot run without the oxygen requiring electron transport chain. Okay. Glycolysis, though, does not require oxygen, and so we call it an anaerobic. Remember, I've taught you guys multiple times that when you see the letter A or AN at the beginning of a word, like abiotic, aphotic, that means it's none. Okay, so no oxygen, non-oxygen, and aerobic, no oxygen, does not require oxygen. Okay, most things in this world are aerobic. Most things require oxygen. Most organisms require oxygen at some point. Even plants, because what does a plant breathe in? Carbon dioxide. It still has oxygen in it. There's a few places that organisms thrive in the anaerobic area. That's going to be things that are like deep underground where oxygen can't get to, things like that. Okay. So even though glycolysis is anaerobic, it is considered part of cellular respiration because its final products are key reactants for our next two aerobic phases. So remember, glycolysis creates 10% of our energy, but 90% of it is called the pyruvic acid, and that is what actually is used in the aerobic processes of Krebs cycle and electron transport chain. So we consider that part of this process because it's what gives us our starting point, if that makes sense. So I told you that cellular respiration occurs in the mitochondria, but that's not the full truth. Krebs cycle and electron transport chain occur in the mitochondria, but glycolysis actually occurs in the cytoplasm. Okay. So just like photosynthesis, yes, it all takes place in the chloroplast, but then there's two parts of the chloroplast, the thylakoids and the stroma. This takes place in the cytoplasm and then finishes off in the mitochondria. If no oxygen is available, fermentation takes place. And yes, you've probably heard of fermentation in the process of making various things, like wine. But fermentation actually takes place in your body as well. We will learn about that, like I said, a little bit later. But it actually, have you guys ever ran or played basketball or soccer and your legs just start burning? That is fermentation. That is the making, or that is your lactic acid that's like leaching out because you are not getting enough oxygen into your body to do what you're requiring the body to do. And so it actually starts doing this fermentation. And we'll talk about that. That's lactic acid fermentation. And we'll briefly talk about that after we're done with cellular respiration. So let's take a look. We are on page 313. There's an image there, a global balance there, about kind of comparing photosynthesis and cellular respiration. So cellular respiration balances out photosynthesis. It's this beautiful process where photosynthesis is the process that deposits energy, And cellular respiration is the process that withdraws energy. So photosynthesis removes carbon dioxide from the atmosphere, and cellular respiration puts it back. Photosynthesis releases oxygen into the atmosphere, and cellular respiration uses that oxygen to release energy from food. So, this is why, even though so many living things use oxygen, we don't run out of it because it's a cycle. We've learned about the cycles of nature. Remember, we learned about the carbon cycle, we learned about the water cycle, we learned about the nitrogen cycle, and just briefly the phosphorus cycle. But we never learned about an oxygen cycle because oxygen is attached to almost everything. It's attached to nitrogen, it's attached to carbon, it's attached to water. So it's attached to hydrogen to make water. So oxygen is so important, but it is cycled through. So we never run out of it. Because remember that very basic concept of science that nothing is created, matter is not created or destroyed, it is simply changed. And so you can see here that I'm not destroying oxygen just because cellular respiration withdraws that. Doesn't mean that we're using it and it's consumed. It just means that we're changing it into something else. So chemically, the process of photosynthesis uses water and carbon dioxide, which both have oxygen, as raw materials, and they synthesize carbohydrates. What's in a carbohydrate? Oxygen. C6H12O6 is glucose. If I change those numbers, I get a different kind of carbohydrate. But this is the one we're talking about. I used carbon dioxide and water, which was carbon, oxygen, hydrogen, and oxygen, and I simply rearranged the molecules. I broke their bonds and I put them back together in a whole new beautiful way. And I now have glucose and carbon dioxide. Okay, it's wonderful. So the global balance between cellular respiration and photosynthesis is essential to maintaining Earth as a living planet. We need plants. We also need organisms like animals because we balance each other out. Okay? Could plants live without people? Probably. Obviously, plants can live without people. But we need some other organisms to be breathing out oxygen so the plants can be using it later. Okay, so let's take a little bit deeper look into these three processes. So, food burns. It's true that a lot of common foods like apples and bananas and ground beef have too much water in them to actually make them burn. Like you couldn't light an apple on fire or a banana on fire. That doesn't seem to work very well, right? Because there's a lot of water in there. But foods with a little bit of water, like sugar, like you could actually light a sugar cube on fire. Okay, you could light flour on fire. You could definitely light cooking oil on fire. Don't do that. That's very dangerous. Please do not set cooking oil on fire because you can't put it out with water. That makes it worse. I hope you all know that. Okay, but in fact, things like wheat grain contains both carbohydrates and proteins and is so flammable that it will actually like almost spontaneously erupt into flames. Okay, so grain elevators, you may probably not around here as much, but like if you live in the Great Plains and they have these huge grain elevators, it is pretty common sometimes for those elevators to just catch on fire because the stuff in there is so highly flammable. Okay, so there's lots of energy available in the food that we eat. But how does our cell get the energy without catching fire or blowing up? Well, it's because we only get the energy little bits at a time. We don't want to take it too much or some bad things might happen. So, the first set of reactions in cellular respiration is referred to as glycolysis. And it literally means sugar breaking, the breaking of sugar. Because glyco, what word does that look like? Glucose. You kind of see that word. Glycogen. Glycogen is a really cool biomolecule that you learned about in health class last year. So it's a breaking sugar. Okay. So during glycolysis, one molecule of glucose, which remember has 6 sugars, 12 hydrogens, and 6 oxygens. Okay, that's what that has. Which is a 6 carbon compound, tells us that in the equation, is transformed into 2 molecules of the 3 carbon compound we refer to as pyruvic acid. Okay, pyruvic acid. If you happen to look on page 315, there's a really nice, beautiful drawing figure of this. You see the 6 carbon come in. Now, obviously, the hydrogens and oxygens are still there right now. We're just not paying attention to them right now. We're really focusing on our sugars right this second, our carbon. We'll figure out what happens with the hydrogens and oxygens in a minute. Don't think they've disappeared. They're still there. But we're just focusing on our sugar right now. So this says 6C sugar. That's 6 carbon sugar. There are other carbon sugars. The 5 carbon sugar should sound really familiar. That's called deoxyribose, and ribose is what makes your DNA and your RNA. Okay, and it's also in ATP, which we'll talk about in a little bit. Okay, so this is our 6 carbon sugar right there. And then it actually uses ATP. So look, we're using some energy. See that? It's using 2. Okay, and it's splitting my 6 carbon sugar into 2 different molecules of 3 carbon sugars, because if you take 6 and divide it by 2, you get 3. So I have 3 carbon sugars here. Okay, then we take NAD, okay, and we produce NATP, and we produce NADH plus. Okay, and we get, so this is what happens. We have 3 sugar phosphate. So we have the phosphate from here, and so on. Okay, so we'll talk a little bit more about that in a second as we go through each of those steps. So even though glycolysis is an energy releasing process, the cells need to put in a little energy to get things going. Okay, so even though it produces ATP, it had to take in some ATP first to get it going. Okay, so as the pathway begins, 2 ATP molecules are used up. Okay, so remember, we learned earlier that photosynthesis and cellular respiration were kind of used as withdrawals, deposits, and withdrawals, right? So similarly, the 2 ATP molecule used at the onset of glycolysis is like an investment. It's like you putting a little bit into the bank account. You have to open a bank account and put money in there to make your money grow, right? You can't just open a bank account and leave it empty and expect money to show up. That's not how that works. You have to actually deposit something, and then after time, and depending on what kind of bank account you have, you get more money. You get money that shows up. It's called interest. Hopefully learn about that in math at some point in your life. The same process. You have to put 2 ATP into the process to get it started. Okay. Once glycolysis gets going, it actually produces 4 ATP. So look, we just doubled. You gave me 2 and I give back 4. Okay, so the 2 ATP molecules are put into the process and 4 ATP molecules are produced. You see that? It's like interest on your bank account. So we get a net gain of 2 ATP. So a net gain, which means that it's the total gain, because we put in 2, we got 4. So it's like 4 minus 2, and I'm only left with 2. You don't say I gained 4 because you're discounting that 2 you put in the beginning. NADH. This probably looks a little familiar because in cellular respiration we have the NADPH, right? And that is because this is another, it's the exact same, it's not the exact same because there's no P attached, but it's still an electron carrier. It carries and accepts those high energy electrons and the hydrogen ion that we get in our water, that we got from water. Okay. This molecule is known as NADH and holds the electrons until they can be transferred to other molecules. So it takes on an H plus, a hydrogen ion, and it takes on 2 high energy electrons. Okay, and it carries them around just like your pan is carrying your hot food around. You can't carry your hot food around in your hands, you'll burn yourself. It's the same process here. And these high energy electrons can be used to produce even more ATP molecules. Okay. In the presence, let's just say presence, not process, presence of oxygen. So what are some advantages of glycolysis? Why is this important? So in the process of glycolysis, 4 ATP molecules are synthesized from 4 ADP molecules. Remember, ATP is triphosphate, ADP is diphosphate. I take a phosphate, I bond it to my other 2 phosphates, and I have put energy into the bond so that when I break that bond and I take ATP back to ADP, I have energy. Energy is the ability to do work. Okay. So remember, because we used 2 at the beginning and we only have a net gain of 2. Okay. So although the energy yield is really small, this happens really quickly. Okay. It is so fast that cells can produce thousands of ATP molecules in milliseconds. That's faster than a second. It's a fraction of a second. It can produce thousands of ATP. Okay, it's crazy. So this is a really big advantage to cells when we need a real sudden increase of energy. But besides speed, another advantage is that it does not require oxygen. This means that glycolysis can quickly supply chemical energy to cells when oxygen is not available. But when oxygen is available, that pyruvic acid and the NADH outputs that are generated become the inputs for our next 2 processes. So when we don't have a lot of oxygen, our body can be doing a lot of glycolysis. And as soon as we start to get more oxygen, bam, our bodies can start to finish the process of cellular respiration. Okay, we are on page 317. There's a big, beautiful diagram of the Krebs cycle right there. You're like, wow, that looks really complicated. It looks complicated, but we're going to break it down and try to be as simple as possible. So, in the presence of oxygen, the pyruvic acid that was produced in pyruvic. Sorry, I cannot talk today. In the presence of oxygen, the pyruvic acid that was produced in glycolysis passes on to the next stage of cellular respiration. So it's going to go from the cytoplasm into the membranes of mitochondria. Now remember, mitochondria has an outer membrane that's like the organelle membrane, and then it has these little folded membranes inside. It's the inner mitochondrial membrane. Krebs cycle takes place inside the inner, inside on the other side of the inner mitochondrial membrane. So during the Krebs cycle, pyruvic acid is broken down into carbon dioxide in a series of energy extracting reactions. Multiple reactions are going to take place in Krebs cycle. Multiple. Okay. You see this? So really, I promise you are going to want to have your textbooks open to 317 because you are going to need to see this as I explain this. Okay. It is something where a visual aid is really, really going to be beneficial. Okay. So citric acid is the first compound formed in the series of reactions. The Krebs cycle is also known as the citric acid cycle. Okay. So during the Krebs cycle, pyruvic acid from glycolysis is used to make carbon dioxide, NADH, ATP, and FADH2. Okay. Because glycolysis produces 2 molecules of pyruvic acid from every single glucose molecule that enters glycolysis, okay, the Krebs cycle turns, quotations around the turns, twice. Okay. Turns twice for each glucose molecule that enters glycolysis. Okay. One molecule of glucose was split into 3 carbons, sugars, which are called pyruvic acid. Sorry, not sugars, 3 carbon compounds, referred to as pyruvic acid. So the Krebs cycle does that twice for every molecule of sugar, of glucose. It only turns once per pyruvic acid. Okay, so again, the 10-4 interactivity. I'm going to try to assign these interactivities as not like an assignment that you have to do, but as an interactivity for you to do in case you can't access your online textbook. Okay. The matrix, it's not a fun word, is the innermost compartment of the mitochondrion. Mitochondrion is singular for mitochondria, and is where Krebs cycle actually is occurring. So citric acid production. So the Krebs cycle begins when the pyruvic acid produced by glycolysis passes through the 2 membranes of the mitochondrion and into the matrix. So that's a fun word. The matrix, not like the movie, is the innermost compartment of the mitochondrion and the site of the Krebs cycle reactions. Once inside the matrix, you have 1 carbon atom from pyruvic acid is split off. Do you see that? Again, follow with me. Please, please, please have your textbooks open to page 317. Okay, so look, we're at the so you got your 3 carbon molecules outside. They move inside, okay? Then then this happens, 1 carbon atom from pyruvic acid is split off to produce carbon dioxide. Do you see that? Okay. Now remember, don't forget, there are still oxygens and hydrogens attached to these molecules. Okay, so these oxygens didn't appear from nowhere. Okay, they were already there, attached. They just pulled those 2 oxygens off of the molecule. Okay. And so now, and 2 high energy electrons are passed to NAD plus to produce NADH. Because listen, let's think about this. Let's think about this for a second. Where did that hydrogen come from? The glucose molecule. Remember? H12, C6, H12, O6. So took that hydrogen, boom, and it took it out of the molecule. Bonds were broken, and it attached it to NAD plus. But NAD plus and H plus, it needs to have 2 negatives. So it needs to have 2 electrons attached to that because positive things don't like each other, right? Okay. Let's keep going. The other 2 carbon atoms from pyruvic acid rearrange to form acetic acid. So they just rearrange to form something else, which is joined to a compound called coenzyme A. Look at that. See those little coenzyme A right there? And they're going to attach to the acid that was made. Okay. to the acetic acid that was made. The resulting molecule is called acetyl-CoA. I don't know if you pronounce it any other way. So then we have acetyl-CoA. Okay. The resulting molecule is the acetyl-CoA. Then, as the Krebs cycle begins, acetyl-CoA hands off that carbon acetyl group, that 2-carbon acetyl group, to a 4-carbon molecule already present in the cycle. And you're like, where did that 4-carbon come from? Look at the end of the blue arrows. Look, it's left. Okay. And then it attaches back to our carbon molecule, but now I have citric acid. You're like, wait a minute, we started off with 6 carbons and it was glucose. Now I have 6 carbons and it's citric acid. What happened? Well, I don't have the same amount of hydrogens and oxygens I did at the beginning, do I? Remember, every living thing on the planet is made out of those elements behind you, the back of my room. The difference is simply the number of molecules or the number of atoms in there, okay? So citric acid does not have the same genetic makeup as glucose. Okay. See if I can find the actual makeup of citric acid. Citric acid is C6H8O7. Okay. So it's a little different, not the same as carbon dioxide, but it has the same 6 carbons. Okay. So anyways, let's keep going around. Okay. So now we have citric acid that's been made. So let's review this process. Glycolysis breaks my glucose molecule into 2. And again, we're focusing mostly on the carbon part of it, but keep in the back of your mind that there are still hydrogens and oxygens attached to these. Okay. They enter from glycolysis happens in the cytoplasm. They enter into the matrix and the Krebs cycle begins. There are 2 turns of the Krebs cycle, but right before they get to the Krebs cycle, they have to actually kind of get prepped. So we have our 3-carbon sugar, our pyruvic acid. A carbon dioxide is taken away. So one of our carbons goes away, along with some oxygens. Okay. Then we are left, also at this point, a hydrogen is taken away, and 2 electrons are taken away. Okay. So those are gone. Okay. Then we are left with a 2-carbon compound. This 2-carbon compound joins. So this is acetic acid right there. This 2-carbon compound is acetic acid. It joins with this enzyme called CoA. It's a coenzyme A. It's shortened as CoA. And it joins, and now it forms acetyl-CoA. Acetyl coenzyme A. Find what that is. Okay. Now it is joined with a 4-carbon compound that's just been hanging out in the cell. And now I have what's called citric acid. And now the Krebs cycle is going to start turning. Okay. So through a series of many, many reactions, let's follow the reactions that are occurring here on page 317, figure 10.4. So first, look at the 6 carbon atoms in citric acid. 1 is removed. Okay, see that? And it's removed as what? Carbon dioxide. What do we breathe out? Carbon dioxide. This is where the carbon dioxide is made. It is made in our cells, passed into our blood, into our lungs, and out our noses and our mouths. Okay, but our lungs don't make carbon dioxide. Our blood doesn't make the carbon dioxide. This process makes the carbon dioxide, and it makes its way out of us. Okay, this happens twice. So one carbon is taken away to make CO2. That happens 2 times. Okay. The reason this is called Krebs cycle is because this 4 compound that is made, this 4 sugar compound that is made, or sorry, 4 carbon compound that is made, just gets cycled back up and then eventually becomes part of the process again. See that? See how it just cycles through and on and on and on? Okay. This 4 carbon compound can then start the cycle over again by combining with another acetyl-CoA. Energy is released by the breaking and rearranging of these carbon bonds. Is captured in the forms of ATP, NADH, and FADH2. Okay. So, finally look at the electron carriers, our NAD plus and our FAD, which is flavine adenine dinucleotide. That's a fun word. Flavine adenine dinucleotide. At 5 places in each cycle, electron carriers accept a pair of high energy electrons and a hydrogen ion, changing NAD plus and Fa to NADH and FAD to NAF. Sorry, I cannot talk. To FADH2. Okay. So what happens to these things that are made? Well, think about it. Carbon dioxide, ATP, and these electron carriers. Well, carbon dioxide diffuses out of the mitochondria, out of the cell, into the bloodstream, and then is exhaled, just like I explained. The ATP molecules are very useful. They are becoming immediately available to power all of our cellular activities. And as for the molecules that are the electron carriers, they are used to generate huge amounts of ATP. Okay, so they're there. So let's take a second and count all of those H's. So let's start at the top with our very first one. So the circle is like an oval and it's made into like a starburst, kind of like a starburst shape. That's the starburst shape is our addition of hydrogens, right? So there's 1. All of the arrows, blue arrows, go around to the right side. There's 2. Go down a little bit more. There's 3. Then we come to this FADH2. How many H's do we have? 2. Then we come up again to an H again. So that's another one. So let's count it. 1, 2, 3, 4, 5, 6. 6. 6 hydrogens attached to this one compound of pyruvic acid. And there are 2 molecules of pyruvic acid that come in at a time. So 2 times 6 is 12. How many hydrogens did I start out with in my glucose molecule? 12. Do you see that I didn't destroy anything? Nothing came out of nowhere? That everything was simply rearranged and changed. So you can see where my 6 carbons have gone and come from. And you can also see where all of my hydrogens have gone. How many oxygens do we have in my glucose molecule? 6. How many oxygens do we see breathed out? How many CO2 molecules do you see made? 1, 2, 3, 6. Okay. Because, and you're thinking, well, that doesn't make any sense. because there's a total of 6 carbons or 6 oxygens in glucose. So this is only 6, there should be 12. Well, what do organisms breathe in? O2. If you flip back to your equation of cellular respiration, it is what? C6H12O6 plus 6 O2. So I actually have 18 oxygens. I've only dispelled of 12 so far. So the other 6 we haven't used yet. Does that make sense? Okay. I hate not being there, but I'm really trying to walk you through this the best that I can. So please feel free to watch the video as much as you can. I'm also just trying to make you guys really understand the concept. I want you to memorize this process to a point, but I want you to understand those concepts, the things that matter and stuff is never created or destroyed. It is simply changed. I am simply naturally changing the glucose and the oxygen into other things. And you can see now how the carbon has gone through these processes, where my oxygen has gone and where my hydrogens have gone. I'm just making sure you guys understand that concept, okay? So our last step, turn the page. 319 has our beautiful image of this. The electron transport. So products from both our Krebs cycle and glycolysis feed into this last stage of cellular respiration. Pause for a second. Okay, go back to page 315. Go back to that figure. Do you see where that NADH has an arrow, a dashed arrow saying 2 electron transport chain? That's because this NADH is moving straight from glycolysis, bypassing the Krebs cycle and going straight down to electron transport. If you scroll back to page 312, you'll see that arrow as well. Okay, let's go to 317. Again, do you see every one of those NADHs, those FADH2s, are doing what? Where are they going? They're going to the electron transport chain. What are those things carrying? They are carrying our high energy electrons. Why do we need high energy electrons? Let's look at the electron transport chain and see. But I want you guys to see those dashed arrows. So those weren't just left there. They are still being used in the process. Okay, I want you guys to see that. Because you're like, well, how do we produce water if all of the hydrogens are attached to these high energy electron carriers? You'll see. Okay, so let's follow those arrows along back to electron transport chain. Okay, so remember that glycolysis generates high energy electrons that are passed to NAD plus to form NADH. Those NADH molecules can enter the mitochondria where they join the other NADH and FADH2 molecules that were generated in the Krebs cycle. The electrons are then passed from all those carriers to the electron transport chain. The electrons are coming into play here, okay? So, NADH and FADH2 pass their high energy electrons to the electron transport chain. In eukaryotes, which is what we are, the electron transport chain is composed of a series of electron carriers, just like it was in photosynthesis. Okay, we have these electron carriers that are located in the innermost membrane of the mitochondria. Now, in prokaryotes, the same chain is in the cell membrane. So this, because remember, they don't always have mitochondria. They don't have mitochondria because they don't have organelles. So this takes place in their cell membrane. We have mitochondria. It takes place there. Okay. High energy electrons are passed from one carrier to the next. Okay, you see all those little beautiful blue H positive ions hanging around? Okay, those are important. Those were brought down. That's where we got all of our fun stuff is from. So we got all those is from our other processes. Okay. Sorry, I had a cough for a second. So at the end of the electron transport chain is an enzyme that combines these electrons with hydrogen ions and oxygen to form water. Remember, we still had 6 oxygens left, right? So we are now taking them and creating water that I told you we would be breathing out. So the electrons are powering this, but because the electrons have left the hydrogen ions are able to bond, okay, with oxygens, and then our electrons are given back, and we are making water. Look at that. Isn't that amazing? Amazing. Oxygen is the final electron acceptor of the chain, which is why electron transport is aerobic or oxygen requiring. Oxygen accepts this low energy electrons at the end of the chain. See, they're low energy now. They're not super cool high energy ones anymore. And without it, the electron transport chain cannot function. Every time 2 high energy electrons pass down the electron chain, their energy is used to transport hydrogen ions across the membrane. During electron transport, hydrogen positive ions build up in the intermembrane. So this is between the outer mitochondrial membrane and the inner mitochondrial membrane. This is positive hydrogen party time. Okay, there's a lot of them there, lots of them there. Okay, it is very, very positively charged in there. In turn, that means that the other side becomes negatively charged, so that the membrane itself becomes sort of this biological battery that can power the synthesis, which is the creation of ATP. Okay. So these high energy electrons from NADH and FADH2 are passed from carrier to carrier down the electron transport chain. Water is formed when oxygen accepts the electrons in combination with the hydrogen ions. Energy is generated by the electron transport chain, is used to move these positive ions across the inner mitochondrial membrane and into the intermembrane space. Okay, now look, as NADH gives off its hydrogen ion and it gives off its electron, it goes back to being NAD plus, which means it's going to move back up to be available for the Krebs cycle again. Same with the FAD. Okay, it's going to shed its 2 hydrogens, shed its extra electrons, and going to go back to being FAD. Okay. So you're like, where is ATP made? I'm not seeing this. I'm seeing oxygen being made into water. Let's look at this. So how does a cell use the potential energy from charge differences that have been built up because it's electron transport? That's what we're trying to figure out. How does that work? Just like in photosynthesis, the cell uses a process known as chemoosmosis to produce ATP. The inner mitochondrial membrane contains these enzymes known as ATP synthase. Okay, ATP synthase. That pinkish, reddish thing on the far right. The charge difference across the membrane forces these positive ions through channels inside of these enzymes, and it causes the ATP synthase to spin, causing it to spin. And with every rotation, the enzyme grabs an ADP molecule and attaches a phosphate group to it, producing a ATP. So let's look at that image there. As the hydrogen ion moves from the intermembrane space into the matrix, it forces the ATP synthase to spin. And as it spins, it grabs an ADP and attaches a phosphate to it. Okay? That's what it is. The real beauty of this system is the way in which it couples both the movement of the high energy electrons with the production of the ATP. Every time a pair of high energy electrons moves down that electron transport chain, the energy is used to move hydrogen ions across the membrane. Do you see the arrows going from the matrix to the intermembrane space, the blue arrows, and they're pulling the hydrogen ions across? That's taking energy to do that. Because how do I know that? Because they're going against the gradient. Things don't like to move to areas from low concentration to high concentration, and those hydrogen ions are definitely moving from low concentration to high concentration. Okay. These ions rush back across the membrane with enough force then to spin the ATP synthase, because these molecules want to get back to the matrix, because it is way too crowded for them in the intermembrane zone. So they're going to go across this ATP synthase so fast, they're going to make it spin, which is creating large amounts of ATP. On average, each pair of high energy electrons that moves down the full length of the electron transport chain provides enough energy to produce 3 molecules of ATP that can be used to power cellular activities. So every pair of electrons produces 3 molecules. Okay. It's a lot. So what are we getting here? What is our totals coming out to be? So glycolysis nets just 2 ATP molecules per molecule of glucose. Because remember, it used 2, it made 4, so we have a net of 2. In the presence of oxygen. Well, sorry, without oxygen. Then oxygen shows up and everything changes. Okay. Together, glycolysis, the Krebs cycle, and the electron transport chain release about 36 molecules of ATP per molecule of glucose. That's a lot when you think about that. That's 36 molecules of ATP are made through this process. That's a lot. So there's roughly 36 ATP molecules per glucose molecule versus just 2 ATP molecules in glycolysis. See how that works? So our diets contain much more than just glucose, though. So, of course, that's not really a problem for the cell. Complex carbohydrates are broken down to simple sugars like glucose. Lipids and proteins are broken down into molecules that enter the Krebs cycle or glycolysis at one of several places. Okay, so yes, we don't have to just eat glucose. It may be the most efficient, but it's not the only way to get food into our bodies. Okay. But how efficient is cellular respiration? This 36 ATP molecules generated represent about 36% of the total energy of glucose. Okay, that might not seem like a lot, but it means that the cell is actually more efficient at using food than the engine of a typical car burns gasoline. What happens to the other 64% though? Well, it releases heat. Think about what your body temperature is. Okay, 98.6 degrees Fahrenheit, about, 37 degrees Celsius. Okay. And this is also why you sweat when you exercise, because you're producing a lot more heat. Okay. So that is cellular respiration. I know that seemed like a lot. Felt like I just talked a lot. And now I'm really tired. I want to take a nap. But we're going to do some activities. I'm sure these notes will take you a couple days. Feel free to pause the notes. After you've watched it in class, feel free to watch the notes as many times as you need. Pause them if you need. And just really, really work on the activities. I said we will briefly go over fermentation. When we get back. Now, yes. Okay, so that is cellular respiration. I hope a lot of that makes sense. I know it's probably a little confusing. I try to walk you through a lot of it. Yeah, so if you have any questions, make sure that you email me, okay? So I will talk to you guys later. Sorry, I'm trying to stop this real fast.